Yeah, well, I'd like to say thanks very much for the kind invitation to speak, and it's really great to see so many old friends again. Now, the last talk was about something that's just recently been done. This talk is going to point towards something I wished I knew how to do, but I have a feeling discussion with folks here could be very useful for that. So it's about data integration. Before I go there, I'll point out I call my research area object-oriented data analysis, and in fact, I published a book on that recently with Ian Dryden. So what is object-oriented data analysis? Well, I explain this by asking you to think about the atom of any type of data analysis. If we organize this by courses, I actually teach elementary statistics courses where atoms are numbers. I try to get students to understand variation in numbers. More advanced students will take multivariate analysis where the atoms are vectors. We've heard a bit of functional data analysis where the atoms are curves. Object-oriented data analysis just continues this in the direction of data objects, which are more general things, much of what we've been hearing about here. So some classical examples are curves as data objects. So functional data analysis is just a special case. If you work with chemical spectra, they need to be non-negative, which introduces a lot of interesting aspects. I do a lot of work with various types of images. I'll show you some today. I don't do it within the image. I think about the image as the atom, and I have a population of images to think about. I also care a lot about shape analysis, and I'll be heading there later in the talk. Another type of data objects is trees or graphs in this theory of network. Again, I don't work within a given graph, but I take a population of graphs. So those are my data objects. You can also do other types of things as well. Okay, so I've been thinking about this abstractly for a while, and I've noticed a very important concept in this field is what I call modes of variation. This is going to be important for later in the talk, so let me describe it carefully. If you've done principal component analysis, you've been constructing modes of variation. So these are fundamental to exploratory data analysis. Oops, what's going on here? Something's not right. There we go. Okay, yeah. So what is a mode of variation? Well, I take it to be just a set of elements in the object space, which is one-dimensional in some sense and tells us something about the variation of the data objects in the population there. So in the case of just simple PCA, there's actually several interesting modes of variation, even in a PCA. One of those is what you think about typically. You think about a point cloud, find the direction of maximal variation from the mean, look at the projections. Those projections you get are a one-dimensional set, and so that's a mode of variation. But you could also take the entire approximating line, not just the projections, and in some cases you actually even want to discretize that. So let me illustrate that in a simple example here. What am I doing? Something's not working quite right. Okay, so here's just a simple toy two-dimensional set of data. Let's think about doing a PCA. You're going to stand at the mean, look in all directions, find the one that maximizes. Then when you project, those projections on the line are a one-dimensional set of data, so that's one example of a mode. But you could take the whole line if you want, and you can even discretize that. Oops, somehow that's not going well. Okay, you can discretize that into an equally spaced grid. Why would you want to do that? Well, let me illustrate that with an example from shape analysis. Did I do this right? Yeah. And so I'm going to work with a set of three-dimensional shapes, do a principal geodesic analysis on that. There it pops. Okay. So these are shapes of objects. I'm not going to go into details here, but you can see there's actually three of them. It has to do with radiation treatment of prostate cancer. And what we're thinking about is variation of the shapes within one person over time. And so we've done a principal geodesic analysis. These shapes naturally lie in a curved manifold. Think about a direction of maximal variation, represent that by a geodesic, stand at the mean, go forward by 2 standard deviations, go backwards by 2 standard deviations. I'm going to jump back and forth between those, and if I'm lucky, yeah, that's working. Okay. Okay, so notice the one on the left is the first principal geodesic, and you can very clearly see jumping back and forth along this mode, you can clearly see that's about an up and down type of variation. The second one is a twisting thing, and the third thing is about the yellow one getting bigger and smaller. Okay, so modes of variation, that's kind of the base idea, but of course I want to take this much further. Principal components is all about directions that maximize variation. You can think about maximizing other things. If maybe you have a couple of different classes, you could think classification here. You could think about a mode of variation that separates your classes. One I like for this is called distance-weighted discrimination. I could go into a talk on this, but I'm just going to remark it's an improvement of the support vector machine in high dimensions. So same set of data here. You might notice I have X's and pluses. The principal component direction goes along like that. It doesn't tell me about the distinction between the X's and the pluses. I want something that points more in that direction. So that also is a one-dimensional subset, which tells me about my variation. And so notice now if I project, I have a direction which really separates the classes. Okay, so I could again take the projections, or I could take the continuous line, or again, I could take something equally spaced. Here's one of my favorite examples of that. Here my data objects are images. And of course, all I'm doing is taking all the pixels, putting them into one long vector, working in the big vector space. These are actually student ID photos from the University of Carlos III in Madrid. And you might have noticed I've shown you a bunch of males and then I've shown you a bunch of females. Now what I want to do is kind of get a feeling for the difference between Spanish males and females by taking these data points in this high dimensional space. I'm going to project on the DWD direction. Here are the projections. Notice this direction separates very nicely the males from the females. And then what I'm going to do is I'm going to march along this direction vector. As I do show, I'll show you the resulting image. This is nobody's face. It's just the point in the space. I'm going to march along there, and you'll see the difference between males and females. There it goes. Okay, so you can actually watch that face evolve in real time. This is the advantage of equal spacing. If I try to look at projections, it herks and jerks, but equally spaced is very nice. There seems to be some perceptual theory that says average is most beautiful. You can think about that. Now we're getting towards the women that probably shouldn't wear men's clothing. The more androgynous Spaniard. There's the more effeminate Spanish male. Then he heads off and gets a bit more macho there. I'm not going to continue, but now you're seeing the value of looking at the modes of variation in an equally spaced way. Okay, oops, let's see, where am I? Yeah. Okay, so now let's start talking about organizational model here. So with a single set of data, everybody's been talking about this. Machine learning people like to call this tabular data, seems to be a fashion. I organize my data in a particular way. I like my data objects to be columns. Why? Because I first learned about vectors in linear algebra class, and vectors were columns. But there's another reason this is very common organization in the bioinformatics world. Okay, so columns are going to be data objects for me. This is some distinction with most statisticians who like their rows to be their data objects. But I like columns, so I'm going to do that. I'm going to use the notation D for number of variables or features, not P, but D for dimension. N is going to be the number of cases here. And I'm going to assume most of my data objects are mean-centered. Now, what is mean-centered? Well, there's two ways you can think about it. One is you can look at every row and take the mean of the numbers and subtract that. You could call it row centering. But I like my data objects to be columns. I want to focus on columns. So I'm going to think of taking the column mean. And so I'm going to call this column object mean-centered. Anyway, that's going to be an assumption for what I'm going to do here. Okay, now let's take a little bit deeper look at the mathematics of principal component analysis. A really nice way to do it is to take the centered data matrix and write it as a sum of rank 1 matrices here. So my notation is U is going to be the kth loadings vector that lives in the d-dimensional space. Sk are singular values. The V's are the n-dimensional vectors. Both the U's and V's are unit vectors. They have length 1. They can be thought of as directions. And the important thing to notice is that matrix product is rank 1 components who express X as a sum of rank 1 components. If R is the rank, that's actually an equality. If people want to do dimension reduction, they might take a smaller R. And just terminology here, the loadings vector determines the direction in the space. You got the point cloud. That direction is given by U. That's a unit vector. And the projection coefficients are given by the scale-free V multiplied by the singular value U. So I put the magnitude in with the scores, not with the loadings. That'll become important in a moment. Okay, so this, of course, is the key to modes of variation here. The projections onto the direction vector give those modes. Okay, so let's see. That's traditional data organized as a single data block. Now I want to head in the direction of multiple blocks of data. And so I'll point out that an increasingly common case is you have not just one data block, but maybe you've got several, and they're going to be from the same cases. This is the key here. So there's a lot of people out there doing this, and there's a lot of synonyms. I like the term multi-data block. Some people will say data fusion, others will say data integration. Machine learning, they kind of like multi-view these days, but for me, view is something I do to data, so I'm not that fond of it. And other people will talk about functional factor models. Okay, well, for me, I'm a person who does things based on motivation. My motivation for DIVAS is cancer research. Cancer is a disease I take personally, so I engage in quite a lot of that. So when I started working with these guys in cancer, it was all about microarrays for gene expression. Microarrays are a very powerful technology where you measure the activity of every gene in the tissue sample. And there were over-advertisers at that time. You hear a lot about AI and how wonderful it's all going to be. Well, the over-advertisers in 2000 were saying, oh, this technology is so powerful, we're surely going to cure cancer. Well, of course, that really hasn't happened. But we care a lot about it, or at least we used to back when we were funding scientific research. Anyway, in those days, we had a lot of money. And so one response is to measure a lot more stuff. Major effort of that type was called the Cancer Genome Atlas. And the idea there was to say, well, we measured a lot with gene expression. Why don't we measure a lot more stuff? And so the technology's there. So the Cancer Genome Atlas had about a dozen different types of measurement. And all made on the same people. And they also looked at multiple cancer types, a very big study. And my colleagues, some of my cancer colleagues were deeply engaged in this. So I've been watching all this happen and thinking a lot about, well, gee, how are we going to analyze data in this kind of situation? So this is something I paid a lot of attention to research-wise. So an important method we developed before DIVAS was called JIVE. JIVE is an acronym for Joint and Individual Variation Explained. So let me give a quick introduction. There's a couple of generations I'm just going to sort of skip through. I show you some of the people. The important thing to realize is how this methodology is organized. So the idea is you got multiple matrices here. Those are the data types or the blocks. And the key is you're going to have common columns as data objects. So you might measure gene expression and this and that and some other stuff. But it's all measurements on the same people. So what are the data objects? Well, it's really the union of these different vectors. Now, you could, of course, just concatenate them and put them into PCA, but there's all sorts of challenges to doing that. But the most important aspect is you really care about understanding how these things work together and how they work separately, which doesn't come out from a concatenated analysis. Okay, so just for simplicity, I'm going to consider just the first two blocks here, just to keep things easy. Notation, like question.
So you're doing joint SVD like 2000?
I'm not going to do SVD. I'm going to do a different operation here.
My question is, you mentioned that you have like 2000 microarray gene. So it's
for example.
So it's 2000 genes or 2000 microarray?
A microarray is a set of 2000 genes. So one microarray gives you 2000 genes. Yeah, right. Microarray is ancient technology, but I won't get into that today. Okay, so we do certainly allow different numbers of features that we're going to measure here, different dimensions. They can be much different, and in fact, they often are in this world. Okay, N is still going to be just the number of people or samples or whatever you like to call those. Okay, so now for 2+ data blocks, the data objects are going to be the unions of the columns. And the game here is I want to explore and quantify the variation that's present. So I'm going to think about modes of variation as in what we did in PCA, but there's going to be some twists on what those modes are here. Okay. So first, I'm going to insist on joint modes of variation. What do I mean by a joint mode of variation? Well, the idea of a joint mode of variation is I'm going to insist that the scores are common. So I'm going to think of rank 1 approximations here, but my rank 1 approximations have the fact that the v vectors are going to be the same, or at least in the same direction. Okay, now you might say, oh, isn't this kind of like canonical correlation analysis? Canonical correlation analysis, you approach this problem by looking at directions here, looking at directions there, try to maximize the correlation between the scores. That can be done by an SVD, a particular type of SVD, but I'm not going to do that. I'm going to insist that the scores are actually the same, not as close as possible, but the same. Okay, so that's a somewhat different thing, and I'll show you why. So those scores, think of that as signatures among the cases, is a good interpretation here. And notice I'm not focusing on the direction vectors, which is what PCA does. I'm focusing on the scores. It's really a dual approach that I'm taking here. Okay, and again, not maximal correlation as in CCA. Okay, so the key idea here is we're working in a really different space. Instead of working the d-dimensional space, I'm going to work in the n-dimensional space. In the machine learning world, they often talk about the dual version of the covariance matrix being the Gram matrix. That's essentially what we're doing. And you might have noticed Jigong did exactly this type of thing in his analysis yesterday. Okay, now how do we find these common scores? Well, for this we use an interesting twist on linear algebra called principal angle analysis. So principal angle analysis was developed as a tool in linear algebra simply to understand relationships of subspaces in a Euclidean space. So I'm going to think about the Euclidean space Rn. Think about a couple of subspaces and how do we characterize how they relate to each other? Principal angle analysis says, well, let's look over all directions in one space, look over all directions in the other space, try to find the minimal angle. If there's an intersection, as you can see in this picture, that first principal angle will be 0. So intersections are characterized by a 0 direction. And so that's the first principal angle. And then after that, just look orthogonal to that. So take the subspace generated by the two vectors you find, minimize the angle over the orthogonal complement, and then you get a second angle. And of course, you can do this through all the way through n. And that set of angles nicely characterizes how they relate to each other. So a nice feature of this is there are interesting linear algebraic ways to calculate this. The one we use is you just take orthonormal basis matrices, concatenate them, and do an SVD of that. Then essentially at one end of the eigenanalysis, you get the one vector, at the other you get the other vector, and the singular value reflects the cosine of the angle. Okay, so now we have a way of finding the vectors that are closest. Now we think about an underlying model where they're the same. Again, key concept, linear algebra happens in Rn. That's why I put an n there. Okay, so now we can do that. Oops. Going the wrong way. Okay, so now imagine we've done this. Now our model is that we believe our data are generated from something where the scores are the same, but when you add noise, that's not going to happen. So we need to get back to that direction. So the way we do it is we take the two vectors that we get out of the principal angle analysis and then basically average them. So we'll just take the simple resultant mean here. Okay, so now we know how to compute common scores. Now to get the full mode, okay, so we now have the idea of common scores. Let me get this right. Yeah, now what about individual? So the next thing we need to do is define individual. Well, the whole reason we're coming up with this common scores is this enables us to get a well-defined notion of individual variation. So our notion is individual modes of variation are those that are orthogonal to the joint. And so because we have this joint, we have a notion of orthogonality, we can define that. Now we're not going to insist that the individuals are orthogonal to each other, but we do insist they're not collinear. But other than that, they can pretty much fall where they want. Okay, again, the linear algebra is happening in the n-dimensional space. Okay, so the advantage of this approach, again, is we get a rigorous definition of what's meant by individual variation. Okay, well, now I've told you what the scores are. We want the whole modes of variation, so we have to be able to talk about the rank 1 matrices. For this, we need the loadings vectors as well. And so now we compute the loadings kind of in the inverse of what's done in PCA. In PCA, you can think about taking that direction vector and projecting using inner products to get the scores. Here we're going to go the opposite way. We start with the scores, we project in the n-dimensional space, and those projections, kind of with the opposite inner product, are going to give us the loadings vectors. And we can do that for the joint modes, and we can do it for the individual modes. Okay, so notice a kind of a fun little counterpoint here between PCA and DIVAS. In PCA, we took the data matrix, projected onto the loading direction to get the scores. With DIVAS, we take the data matrix, project onto the scores to get the loadings. One little tricky thing here. In PCA, scores and loadings are all orthogonal to each other. That no longer holds here. There's no guarantee of orthogonality when we do these projections. Okay, so just to contrast this again, thinking about these low-rank approximations, everything's using a low-rank approximation with PCA. So there are different groupings of the singular values. So these expressions, the only difference is where does the S go? Goes in different directions there. In PCA, we're sort of thinking about the U's as the directions, and so therefore it makes sense to group the singular values with the projection coefficients. But in DIVAS, we want to keep the unit-free directions. So what we do is take the singular values, put those together with the U's instead. So it's natural because the scaling depends on the data block. It's natural to put it in with a particular data block. We want something unit-free to be common across. So that gives an easy way to represent the scores there. All right, well, gosh, I've gone through all this, haven't done any real data yet. Let's take a look at some real data. So I'm going to look at something called the Carolina Breast Cancer Study. It's a big database collected by my university. There's a lot of very diverse people that work with me here on this. I'm very proud of the fact that I've got both pathologists and genomics people as co-authors on this. My former student, Eden Carmichael, did all of this work. And so this has to do with pathology. What's pathology? Most types of cancer are diagnosed by taking a tissue sample, it's stained, an expert called a pathologist looks at this, and based on his vast knowledge and experience, he makes a call on your cancer. That's very bad cancer. Oh, that's not cancerous tissue at all. The tissue is stained with a couple of colors called hematoxylin purple, stick to the nuclear material. Eosin is pink, sticks to the other stuff. And of course, it's a natural place to think about doing some image analysis instead of having your expert work on it. And so that's the kind of thing I want to take a look at here. And so the analysis goal I'm going to look at, I'm doing data integration. I'm going to integrate the images and the genomics here. Why the images? Well, that's the current diagnostic standard. So certainly makes sense to work with that. Why genomics? Well, genomics is where all the exciting research is happening. There's a lot of deep insight being developed and also new treatments. For example, in breast cancer, I stopped counting in 2020, but at that time, there were already 70 different treatments for breast cancer, a lot of it driven by genomics research. Okay, so now my data integration goal is understand how these things work together and also how they work separately. And so the analysis we did at this time was AGIVE. And so let's have a go at that. So what's the form of the images? Well, the images here were called tissue microarray data. They basically take a piece of tissue, poke it with a needle 1 millimeter in diameter, get a core of tissue, and then they cut it into slices. They take each slice and do different things, but what I'm going to show you here is typically cores drawn from about 4 different areas and then one slice. So we get an image which looks roughly like this. Okay, so that's going to be the data objects on one side. Experimental design, we had a pretty big data set of about 1,000 people here. For each person, we've got 1 to 4 of those tissue microarray cores. And we also have gene expression. So here I'm showing you a picture of a microarray. So this is one person. Each one of these dots represents a gene. And then we have that over all 1,000 people. Okay. So let's see now. What about the genes? Well, the gene set is kind of embarrassingly small here. We're going to work with just 50 genes. The reason is we didn't even use microarray technology here because of the fact that these things are expensive, and with 1,000 of them, it's hard to collect so many. We're going to work with a set of 50 genes known to be very important for breast cancer. This is measured at expression level. This is, as I said, early technology. More recent technology called RNA-seq will give you tens of thousands of genes at a very detailed level, but I'm not going to get into that today. Okay, now what are these 50 genes good for? Well, they're very important at separating subtypes of breast cancer. And in fact, it's my colleague Chuck Peru who discovered the class called Lumino A. Classical treatment for breast cancer is give everybody carbotaxyl chemotherapy, very harsh treatment. And what was known about it was not everybody got any benefit from the chemotherapy, but they didn't know who was who, so they give it to everybody. Well, Chuck discovered these different classes, and the Lumino A's are the people who don't benefit from the chemotherapy. Okay, so now we've got these images, we've got our genes, we've got our images. We need a numerical representation of the images to put it into Jive. So the numerical representation went like this. We used some neural nets to represent our images. Now the problem is neural networks have a very powerful way of doing this type of thing, but the problem is they don't work on such big images. You can see these images are about 2,000 by 2,000. Most neural nets require an input with pixel sizes in the hundreds. So what we did was at this first go, we just randomly cut these up into 100 randomly selected 224 by 224 patches. Now, currently we do things more like tiling, like you put these right next to each other. This was just the first go, so we just did it randomly. Okay, now why 224 by 224? Well, the reason is that that's the image size in a famous neural net called VGG16. So this comes from the world of natural image recognition. Some time ago, Google put together a really big data set called ImageNet. Basically, they took 1,000 classes of natural images. So think things like cars and trucks, dogs and cat, breed of dogs. They had 1,000 different classes. For every class, they had 10,000 labeled images. And then people in the image recognition world would compete with each other and see who could get the best error rate. So over 10 years ago, VGG16 was the winner, so that was considered good at that time. Of course, people have been improving, but we don't keep up with it that well. But anyway, we use this older neural net, and we basically put all of our images into this. Now, we could get a label of dog or cat or whatever, but we don't care about that. So instead, we reach farther back in the process, grab a 512 feature vector. So for each patch, we're going to represent our feature vectors by a single vector. Okay, so that's at the patch level. Now, what about at the core level? Well, we now have 100 different patches at each core. You could think of different ways of connecting those. The simple thing we did first is just average. So we're going to consider just averaging at the core level. Then we got 4 cores per person, so we can average that. So now I have a 512 feature vector either at the patch level, at the core level, or at the person level. And the person level is where the genetics are going to happen. Okay, so now I've got my data set, I've got my 512 vectors for every person, and I've got my gene expression for every person. Let's put all that into Jive and see what we can find. So we found the first joint mode of variation. Now we can look at the scores, but they're not that interesting. It's just kind of a normal population. But if we look at the loadings, that's where the interest happens. So now I need to visualize the loadings. How can I do that here? Well, the problem is it's in this 512 feature space. I don't know anything about what those things mean. So the way I think about this is we got this population, and we found a feature vector whose scores are the same as what we've got in the gene space. And I want to understand intuitively that direction in the image space. So I just do the simple thing of taking the person at one extreme end and look at that person. Okay, so here's our visualization for that. So that person at the extreme end had those 4 cores you can see at the top. Each of those cores had 100 patches. We then took the 16 most extreme patches. Okay, so here's the 16 most extreme patches. That row is I've grayed out the image and then I've colored in just where those patches are so you can see where they came from. Okay, so what are we looking at here? Well, this is the point I lean on my pathology colleagues. And the pathologist looked at that and said, oh yeah, this is fat cells and stroma. Fat cells are a very natural part of normal breast tissue. Stroma is the connective tissue. And so this is basically pretty healthy looking tissue here that came up at one end. What happens at the other end? Well, it's a, oops, didn't go there. There it goes. Very different picture. So what's going on here? Well, clearly it's very visually different. What's the meaning for cancer? Showed this to the pathologist. His first remark was, well, this is highly cellular. What do you mean by cellular? Lots of cancer is about cells rapidly dividing, and rapidly dividing cells can push everything else out of the way. And so that's why you're not seeing any of those other cells. These are just rapidly dividing tumor cells. And he also says they're markedly atypical. So pathologists see a lot of things we just don't see. But basically what's going on is it's almost all nuclei, and the nuclei are all kind of spread out in a very ugly way. So this is a very bad type of cancer here. Okay, so now what we see, what we found on the two ends are pretty healthy and a bad type of cancer going on out there. What's going on with the gene expression? Well, keep in mind, I've found scores that are the same between the images and the gene expression. So here's the gene expression. So these are the 50 genes, and all I'm showing you is the loadings. The blues are the genes that had a positive loading. The reds are the genes with a negative loading, and we've sorted them by absolute value of the loadings. Okay, so I imagine you don't know too much about those genes, but I have good friends who know a lot about them. I showed this to Katie Hoadley, and she got very excited because the blue are what's called high proliferation genes. Proliferation is about cell division. Cancer cells are dividing quickly. When the cells divide, there's certain genes that are very active, and that's the blue ones here. So that made perfect sense. And then what about the reds? Well, the reds are the luminal A. We call the luminal A are the people who should not get the carbotaxol chemotherapy. And so now we've associated what's going on with those genes with what's happening in the images there. Okay, so everybody was very happy that this lesson was very consistent with the images. I can feel time moving on, so I'm going to go a little quick. Here's the second direction. The short story is you're seeing something very different, but the pathologist says it's a bad type of cancer. When you go to the other end, you're seeing something that looks different because there's these red stripes going through there. But those red stripes are, the pathologist says, low nuclear grade, not a bad type of cancer. And the stripes are something called sclerosis. Sclerosis is about tissue damage. And so again, it's a type of cancer versus something healthy looking, although visually it looks a lot different. And that, of course, is what we're doing, is looking visually. Now what about the corresponding gene expression? Well, that's this stuff. Katie says, well, that healthy is luminal subtype genes, so that makes good sense. And she liked this other one because she says, well, those genes are characteristic of the basal subtype. So basal is one particular type of cancer. Now we're heading in the direction of actually understanding how these things look visually different. So then there's also individual modes of variation. First individual gene mode of variation was this. What's going on there? Well, the thing to know about gene expression is they measure these things by amplifying the RNA. And one thing about it, they can amplify a lot, but what they can't control is the amount of amplification. And so what you're seeing here is a very natural amount of amplification effect, and that's individual. It's not related to what's going on in the images. It's just a random lab effect. So that shows up nicely individually, so that made good sense. And we can also look at the at individual modes of variation. Here, I'm not going to put in time on this, but basically you're seeing something visual that looks a lot different, but is not cancer related. So that all makes sense. Okay, so now I want to get to the next step in this, which is a method called DIVAS. So DIVAS is an improvement of JIVE. I have some other collaborators here. And how does DIVAS improve JIVE is the question. Okay, so one aspect is partially shared blocks. So I didn't say anything yet about more than 2 data blocks, but if you do JIVE with more than 2 data blocks, it can handle it, but the modes of variation you're going to find are either joint between everything or individual, again in the sense of orthogonality with the joint. Okay, but once I lay it out like that, you can easily imagine other types of jointness you'd like to do. You might like modes of variation that are joint between the first two, but not the third, and so on. Okay, so this is one aspect of DIVAS. Another aspect of DIVAS is there's a better initial low rank approximation. One of the weakest points of Jive is there's a tuning parameter. You have to come up with a low rank approximation, and that's as slippery as trying to choose the right number of principal components. Yes?
In the previous slide, how do we choose how we do the partial sharing? Is this something that we go into ex ante with?
The answer is we look at all of them and find anything that's there.
So it's data driven?
Oh yeah.
Okay.
Yeah, that's one of the improvements as well. Okay, so Jive has a problem. There's a tuning parameter, and it's as hard as choosing the number of principal components. So I feel like I have a lot of experience. I can kind of look at data and choose it, but I'm really slow to give that to somebody who doesn't know what they're doing because they can easily shoot themselves in the foot. And one of the other big improvements is we do that a lot better now. We've been doing some random matrix theory, Marchenko-Pasteur shrinkage ideas. We've also done something novel called random direction bounds. Not going to put time in on all that stuff. But anyway, that's a big improvement. And I skipped past it, but we also have an iterative search approach, which gets at the question that was just asked. How do you know which one you look at? We actually search through all of them, find all the ones that are there. Okay, so better statistical inference is another major improvement of DIVAS over Jive. Now, just for fun, I'm going to take a look at, do some DIVAS analysis with a multi-block data set from the neuroscience world. So Andy Ackerman is a student who did that. Jungyu Zhang is a colleague who does neuroscience. So we're going to look at data from the Human Connectome project. And so this data set had big data blocks. One is called functional connectivity. So this is where people sit in the fMRI scanner and they're doing some task. And while they're doing it, they look at how different parts of the brain light up with respect to the task being performed. And here they chose to cut the brain up into 87 regions called regions of interest. And then what they think about is basically correlation between each pairs of regions there. Okay, so that's one data block. Another one is structural connectivity. Structural connectivity has to do with the wiring between the brain. They actually literally measure the fibers going between these different regions and essentially measure how many fibers connect up same pairs of regions. Now you'd expect those to be associated because if there's a lot of fibers there's probably a lot of joint activity going on, but they're not exactly the same. Okay, another thing they do is called cognition scores. Here's things like speed and accuracy on the task being performed. It also includes things like delay discounting, sort of personality measures. Now, this was a drug abuse study, so they also looked at drug abuse, self-reported. And then finally, we actually did some genome-wide association data. So this is SNPs. It's basically locations on the DNA, which is a good map of human variation in terms of their genetic plan. Okay, so we had 375 people we were working with here. Now, it's one of these data sets where they post it and pretty much anybody can have it if you're willing to put in enough time to actually apply and all that kind of stuff. But anyway, it's basically publicly open data. So a lot of people have been working with it, but almost all the published studies, they say, well, I know how to associate two, so they look at various pairs. So they've done a lot of pairwise analysis, but we think we're the first to really take this on 5-way. Okay, so we let, in DIVAS, the data determines the interactions instead of just choosing pairs to look at. Okay, so here is our DIVAS diagnostic. And so let's see now, there's a lot of stuff going on. There's all these different ways to pair 5 things up. First thing is, what about a 5-way? Can we find a 5-way mode of variation? Well, we tried it. There was no significant mode. It would not come above the significance level. So that's reflected by this first column. We got all zeros and grays here. Then we tried the 4-way modes of variation. Unfortunately, you can see how the different patterns go. Unfortunately, none of those turned anything up either. We thought we were so great doing 5 ways, but we didn't get that much better than the pairwise. We did get a single 3-way mode of variation here. So we did find one 3-way, so that was okay. All the other modes of variation are pairs. You can see all the different possible pairs here. Okay. Oh, interesting thing is what we do is put the dimension of the DIVAS subspace. So the subspace that spans those two actually is 27 dimensional. So what that's saying is when you compare the function and the structure, because of the fact that the wires determine how much information is passed, there was really a lot of variation there. It's a big space because there's all sorts of ways in which those things can vary together. So that made sense. Okay, unfortunately, okay, then, yeah, so we can look at other pairs and so on. Question?
Quickly, what's that green bar over there on the bottom line? 130, what's that?
Okay, so this is 5-way, 4-way, 3-way in blue, 2-way, this is individual. So these are individual modes of variation. And so, yeah, notice we didn't have any for cognition. Cognition, when we do our low rank approximation, the rank was only 10. So we only had 10 degrees of freedom, and actually that was always associated with something else. 6 of them with function, 3 with structure, 1 with the drug abuse stuff. Okay, so yeah. Cognition and also the survey data of drug abuse is also very small. There's just not that much stuff to look at there. So that was fairly low dimensional. Yeah, surprisingly,
Some kind of
sorry.
section of the brain are you at?
It's the both cortical surface and subcortical structures. So it's parcellated into, I think it was 87 regions.
Okay.
Yeah, so they use some neuroscience knowledge and the free surfer type of representation and things like that. Yeah, every time I talk to somebody, they're doing different parcellations. I don't know exactly how they do it, but
It's very low dimensional, the whole brain.
yeah, but keep in mind we're taking these 87 things and then we're doing is look at all pairs. So it's like 87 paired is the dimensionality we're putting into DIVAS here.
Okay.
So I don't know, big, small, yeah. Anyway, that's the kind of thing I rely on my experts for. Okay, yeah. And there is a lot of genomic variation, which is individual, not surprising because the genomics is a blueprint for the whole organism and everything going on. And so it's not surprising. There's lots of stuff with this individual for the genomics. Okay, now let's start looking at some of the loadings. So for that 3-way mode of variation, here's a plot of the scores, somewhat similar to what I did before, but we're looking at bars. An added feature is we did a hypothesis test for which of these things are significantly different from 0. The ones that are not are grayed out. And so interestingly, there's a whole lot of stuff working here, but it's kind of a, do you have bad drug abuse habits or not is kind of pretty much what that mode is there. So that makes sense that popped up. We also looked at connections between these regions of interest. And so this is a circle going around the circle of the regions of the interest. So these are just parts of the brain. They're kind of labeled with some kind of stuff that makes neuroscience sense. Okay. And we also only showed the significant connections. And so my colleagues were actually able to interpret some of these. So we felt pretty good about that. Okay, so that is DIVAS. This is data integration. Now, I said this is a talk about something I haven't done yet. What I haven't done yet is this type of analysis working with data objects lying on a manifold. So I'd like to do just a bit about data objects lying on a manifold. Fortunately, there's been a lot of talks which have laid the groundwork for this. I think about manifold data, think about manifolds as a surface where you have local charts, to use Stefan's nice term. Of course, the idea of it manifold is smooth in the sense that at each location there's an approximating chart. Sometimes I call that mildly non-Euclidean. People heard me use that term yesterday in a discussion. Choice of manifold, a nice one often is to use the geodesic mean. Also, Frechet mean or Berry center are synonyms for that. Okay, so in fact, I was using a geodesic analysis when I showed you this example previously. And then as people have been talking about, another method for doing this kind of stuff is principal nested spheres analysis. This is an intrinsic analysis. I agree with the previous talk. That's an important way to look at things. Here's how I illustrate principal nested spheres. I'll think about data on the sphere. I'll think about projecting onto a subsphere of one lower dimension. I'll represent that by the gold thing here, nicely parameterized by a slicing plane. Then what you can think about is how close are the data to the subspace. And so we like to do that intrinsically. We like our distances to be geodesics along the surface of the sphere. And then you can think about wiggling around that gold subsphere to get the best fit possible, something like this. Then if you want scores, you can keep the signed residuals as scores. And then, of course, what you can do is go down to the next lower dimension. So basically, those green points in SD now lie in SD minus one sphere, and then you repeat this process. Okay, so that's good. Okay, now what I'd like to think about is modes of variations. I've given you scores, but not necessarily modes of variation. So now imagine we've come down to the S2 level, and so the interesting thing to look at is the best fit S1 in gold. And let's think about modes of variation here. Okay, so something I haven't said anything about is center point. Principal nested spheres does have a center point, and the center point is we'll come down to the S1 mode of variation, and we'll take the Frechet mean on the circle. Notice that's different from the Frechet mean everywhere. 2 minutes, okay. All right, in 2 minutes, I'm going to jump to the end here. I did start a couple minutes late. Anyway, let's just jump to the end. Okay. Yeah. Okay, so there was some fun with data on the torus, which I will skip. If anybody wants to see that, I'm happy to talk about it at some point. Okay, so now what I wished I could do, that I promised you that. Well, the title of the talk was Data Integration via Analysis of Manifolds. So replace the Euclidean subspaces by manifolds is the idea here. So what I'd love to be able to do is figure out how to develop joint and individual modes of variation for data objects lying on manifolds. Okay, so for example, with there's a neuroscience data set I contemplate working with, different from what we had before. The brain function, brain structure are the same as what I had, but now we're going to think about other things. And I have a colleague who thinks it's interesting to look at things like shape of various organs. So I have a colleague in psychiatry who wants to do DIVAS analysis, but what he really cares about things are shapes of various organs, like the shape of the heart, shape of the kidney, shape of the liver. I'm not sure why he wants to do this, but I think he knows what he's talking about, and I anticipate having some fun in actually doing that. So there's real question marks in my mind. Principal nested spheres, we had a lot of success using a backwards approach. Start with a full sphere, come down in dimension iteratively to develop a nested series of approximating subspaces. Well, the problem is in DIVAS, I don't think we can do it backwards because we don't really want to think about a full decomposition. We just want a few modes of variation which somehow naturally come out from a forward analysis. DIVAS intrinsically uses principal angle analysis. Is there a notion of principal angle analysis where you're working on the sphere like this? I don't know. I think so, but I'm really not sure. This is an open question, and I'd love to hear what other people have to say about it. Surely it's going to boil down to some sort of optimization problem. What's the appropriate optimization? Well, again, I really don't know. Okay, so that's my questions. Just to finish up, carry away concepts. Notice it's all based on modes of variation. I think that's really important to so much of what's going on here. They give useful insights, fundamental exploratory tool, and in fact, I find it a common thread across object-oriented data analysis. All right, I'll quit here. Thank you very much.